Okay. These are in a membrane, right? And so outside this membrane, we've got the intermembrane space. So that's the space between the two mitochondrial membranes. This is the matrix of the mitochondria right here. We have over here, we've got NAD plus that gets, oh, sorry, other way. NADH is going to come in and drop off its electrons to make NAD plus. Those electrons will go through complex 1. Some of that energy that goes through will be captured, and we'll pump out 4 protons. Okay, over here then, we're going to inside, pretty much in the membrane, we have a ubiquinol that comes in, a ubiquinone that comes in and becomes ubiquinol. It picks up those electrons, and it's going to move over to complex 3 and drop off those electrons and become ubiquinone again. Does that make sense? Okay. Those electrons are going to go through complex 3. As they go through complex 3, some of that energy, as they're going to go from higher to lower energy, some of that energy will be captured, and we'll pump 4 more protons across the membrane. On the other side here, what we have is we have cytochrome C with an iron in it in the +3 state. That's going to pick up an electron. It's going to become cytochrome C with an iron in the +2 state. It's going to bring that electron over here and it's going to drop it off to complex 4. This has to happen twice because this is a 2-electron process, and cytochrome C can only carry 1 electron in that way. Cytochrome C, I draw it above the membrane here. What is this protein that has this big hydrophobic piece and it kind of floats back and forth? It's got a tail that goes into the membrane. The cytochrome part is outside the membrane. And then those electrons go through complex, those 2 electrons go through, that get transferred over, go through complex 4 here, and 2 protons are pumped, total, not per electron. And then on this end, we have oxygen that comes in and makes a water. Okay? So you should be able to do that, right, in terms of that. But what we were talking about at the end of the last lecture is electrochemistry, right? So we should be able to look at each of these reactions individually. Last lecture, what we did is we took the starting material, starting reaction, the ending reaction, we figured out the change, right? And it was okay for us just to look at what happened at the start and then take what happened at the end to get the total potential change here, which we then also calculated to a free energy. But it's okay to do that because of what? Why do I not need to worry about this one and this one to get the total? Why do I not need to worry about what happens in between? Okay, we've seen this a couple times. Remember, if I've got some process where A goes to B and B goes to C and C goes to D and D goes to E, I'll stop there. But that B and B cancel and C cancel and D cancel, right? We can add all these reactions together and we're going to have these reactions. We have a reaction where ubiquinone becomes ubiquinol, okay? But that ubiquinol then gets converted back to ubiquinone. So we just get the same thing back. Good potential both ways. That makes sense? Okay. I mean, the potential difference would also be the same even if these electrons went from here to here directly. We would have the same difference in potential and the same free energy change. It's just biologically you can't capture that energy, or else I'm assuming that's the way it would work, right? Okay, but let's go and look at some of these. Let's look at some of these reactions individually. Okay, I have in the past asked you a question where, hey, I want you to arrange an electron transport chain where each step is spontaneous, so protons can be pumped. Does that make sense in terms of a question? So how would I arrange if I had these cofactors? If I had this one, and that one, and that one. How would I arrange them so each step was spontaneous? Well, obviously that way. But if I gave you an alternate universe problem, how would that be? We'll do one in a second. But before we do that, let's just take an individual reaction. So when we think about complex, if we wanted to look at one of these, if we talk about complex 1, we have NAD plus, sorry, NADH comes in. Oh, I'm stuck on that. But NADH comes in and makes NAD plus, right? Dumps off its electrons, and over on the other side, we have ubiquinone picks up those electrons to become ubiquinol. Right? There's hydrogen involved too, but they're not really, they're just kind of carriers of things. I'm ignoring the hydrogens to some, I'm not talking about the hydrogens as much. So what's the potential for that reaction? What is the potential change and the free energy change for the reaction above? Okay, so we're going to need this table. Where does the table go? There it is. Let's go ahead and pull this table. Oops. Let's go ahead and pull that table. We'll copy it up here so we can see it. Maybe it will refuse to be copied. All right, so you guys can't see it super well, but we could grab our 2 here, right? Let's make it huge. There, if we make it enormous, what is our potential change for our NAD plus? We've got NADH going to NAD plus, right? What's the potential change for that? 0.315, I agree. Negative or positive? Positive, because we're flipping it, right? So you guys remember 0.315. You got better math. Your brains work better than mine at this point, right? You're better at remembering things. And then we're going to ubiquinol. Where the hell is ubiquinol on here? Excuse me. Here, ubiquinone to ubiquinol, right? Okay, and so that's what? 0.045. Negative. Negative or positive? Negative. Is it? So it's Q goes to QH2, right? So positive, like it is, just the way it reads. All right, so let's go ahead down here. All right, so we have NADH, right? That's a reactant. So when we look at these arrows, I didn't draw this arrow, but when we look at these arrows here, the things at the start of the arrows are reactants. So we have NADH, we have ubiquinone, right? I'm not going to worry about the protons again, but there are 2 electrons on this side, right? Well, actually, there aren't 2 electrons. There's 2 electrons in each, and they're going to cancel. Actually, I should have done a different problem. Oh, well. Oh, well. Should have done one where we had to multiply times 2. But got ubiquinol, right? Plus NAD plus. And so what we said was that NADH going to NAD plus, that was a half reaction potential change of it was positive 0.315. Okay, and then the other one, ubiquinone going to ubiquinol, that was a potential change of, that was 0.045. Okay, great. And so we can add those two together, right, and get the reaction up above. And so our potential change for this reaction, the total potential change for a full reaction, right? We've got these half reactions that we put together to make a full reaction where we have one thing is oxidized and one thing is reduced. It's going to be 0.360 volts, right? So, all right, there we go. All right, and so is that spontaneous? Yes, because it's positive, and because when we talk about potentials, positive is spontaneous. I don't know what the historical reason for that is. I blame physics. It is probably physics' fault, but that's what it is. So then if we want to get the free energy out of that, the free energy change is going to be equal to negative n, how many electrons were transferred, times Faraday's constant, times the change in potential. All right. How many electrons were transferred? 2. Faraday's constant is 96,500 joules per mole. Okay. Yeah, there's not a Kelvin in there. All right, times the potential, or joules per volt mole, right? Because it's going to have to cancel. Times our potential change, which was 0.360 volts. Volts cancel as volts. We're going to end up with joules per mole. I should have a negative sign out there. Yes, you'll be given Faraday's constant. You will not be given the equation. And so what I've got there is, I'm going to convert it to, you guys okay if I convert it to kilojoules in my head? So 64.4, we had three significant figures, kilojoules per mole. Okay, and it's negative. So also spontaneous. And also a ton of, that's a really spontaneous reaction, right? When I look at that, the hydrolysis of ATP is 30. So a lot of energy there. Not all of it gets captured, as we discovered last week, or described last week. Questions? So I want to show you, so if I was to ask you about what's more spontaneous, things go, if you look at this table, there's some things you can do to, if you were given what order would these things be in, or what will spontaneously transfer its electrons to something else, is what do we start out with in the electron transport chain? First reaction. NADH, right? We make NADH. NADH comes in and drops off its electrons to what? Complex 1, but then they go to ubiquinone, goes to ubiquinol. And then those electrons get transferred to cytochrome C. There's two C's. We'll just pick one of them. I don't even know which one it is. Okay. And then they go to oxygen. So what do you see here for every reaction to be spontaneous? The reduction potential gets more positive, right? You start most negative down here, we go more positive. That's because you're flipping these, right? We talk about this is the potential to reduce. So if I flip it, it's the potential to oxidize. We're handing electrons off each time. Questions? Okay. So if I ask you, if I tell you, hey, make an electron transport chain, well, let's do one. Okay. Make an electron transport chain using pyruvate, fumarate, and nitrate. So it's only going to have 3 instead of 4, right? So make an electron transport chain that is spontaneous at each step using pyruvate, fumarate, and nitrate. All right, and so you can use this. I don't know if how well, can you see that well enough? So give me 2 complexes, complex 1 and complex 5. Yeah, because it's not a real electron transport anyway. Or not a real electron transport chain anyway.
Can you start with lactate?
That's a good question. Would I start with lactate? Yes. Where's pyruvate? Right here. I would start with lactate, and I would make pyruvate, right, which would give up 2 electrons to the next complex, which is succinate going to fumarate, and then fumarate. Oh, hold on. No, I'm sorry. Fumarate going to succinate because we're dropping off the electrons, right? And then succinate would go to fumarate to make we take nitrate to nitrite. Okay, so here's how I would do it. I would go, I've got complex 1, I've got complex 5. All right, and so I come in with lactate, right? Lactate is the reduced form of pyruvate. So I take lactate, I bring it in, I get pyruvate. On the other side, I'm going to come in with whatever the oxidized form of my next one is. So I'll move up until I see fumarate. So here we did pyruvate, right? Now we're going to do fumarate. And it would have been a better problem if I hadn't given them to you in order. I realized that I'd done that. But so you'd have to worry about that. So fumarate, and then fumarate goes to make what? How is it? Succinate.
So are you always going from bottom to top?
Yes. If it's specified that each step must be spontaneous.
Okay.
Otherwise, all you'd have to do is put the bottom one at the end and the top one, or the bottom one at the start and the top one at the end would make the whole thing work. But each step might not be spontaneous. And then at this end, we had nitrate going to nitrite.
Do you also need water supply?
You could.
But they're not required to?
What is it? We got nitrate, nitrite. Yeah, I mean, I guess you could include the water. Technically, you'd want to, I mean, you could put the protons for the water up above. I'm playing fast and loose with the protons. I'm not worrying about hydrogens in terms of these things. Yes.
Can you like explain how you know which way like to go? Like
In terms of these arrows.
well, like yeah, like I was thinking you go from negative to entry.
Hmm? Yeah, so that's great. That's a good question. So what we've got here, the question is, how do I know what goes on which end of the arrow, right, or where they go? So we have, for each of these cases here, I'm going to actually erase my purple lines because I think that probably makes it harder to see stuff, right? You saw it as we followed it up. But this is the oxidized form on the left-hand side. This is the reduced form on the other side. So for these complexes, one way to think about it is everything that's on the left-hand side of a complex is going to drop off electrons, right? And everything over on the right-hand side of the complex, the arrow on the right-hand side, is it picking up electrons. So dropping off electrons, we're going to go, I'm going to go ahead and write this on here. So we're going to go from reduced to oxidized, okay? And we're going to go from, once again, this is going to be oxidized to reduced, okay? And this is going to be over here, this is going to be oxidized to reduced. All right, so the oxidized and reduced are bouncing around, but what we're following in terms of these arrows is on this side of a complex, we're going to be oxidized, we pick up electrons, and then we drop them off. And then we pick up electrons. So you're always on this, if you're on this side of a complex there, you're picking up electrons. So they'll oxidize to reduce, whichever way the arrows are drawn, right? I could draw them the other way, but I don't know how the, on the quiz that I've done this on before, I don't remember which way the arrows go. But it's always going to be oxidized to reduced on that. That makes sense? Yeah. How do people feel about that?
I thought it was just like do whatever you see.
You could do that as well, as long as the arrows make it spontaneous. So I agree. If you look at that, it's only going to be spontaneous if they go in that direction. Yes.
So like, so say there's like we have almost like 3 complexes. Where's the point in that? I know it's like left side or dropping off.
Yeah.
So like, where is it cut off in the middle? I guess to switch from dropping off to picking up in the protocol.
If you're saying if we had 3 complexes,
like yeah, like it's just in the middle.
if I added a 3rd complex, I'd need to add another set of reagents. Does that make sense? Yeah. So we have 3 half reactions here for 2 complexes. We have 4 half reactions if we have 3 complexes. Does that answer the question?
Yeah. So is the first one, say if it's 4 different ones?
Yeah.
Is the only one that's going to go from reduced to oxidized the first one?
The only one that only goes from reduced to oxidized, yes, because it's dropping off its electrons at the top of the chain. It has to pick them up someplace else, like the citric acid cycle. Okay. Other questions? And as I've alluded to before, and I'll find some example that I'll cover later in the course. I'm not sure where. Maybe during. I'm not sure where I cover it. I usually do mention it, but there are a bunch of different types of electron transport chains that like bacteria will use depending on their environment. So you can have all sorts of things as your final electron acceptor. In my fish tank, I have bacteria that are doing this. This is their final electron acceptor, okay? And then I have, actually I don't think in your fish tank, because there's oxygen around, you don't have bacteria that make it into nitrogen and bubble it out. That'd be really nice. Then you wouldn't have to change the water so much. Yes?
So if we were to draw like the protons being bumped out of complex 1 and complex 5, it would be 2 protons, 2 protons, and then
I don't know how many protons. It's not based on how many protons are in the reaction.
Okay.
That's a great question. So it's not 2 protons because there's 2 protons here, right? Because in the first complex, right, we've got 1 proton here in this transfer to ubiquinol, but 4 protons get pumped.
So then what's its
It's how much energy there is and how much of that energy is captured. And I'm not going to go into the detail of how those protons are moved, You're welcome to look that up. But some of those protons are actually these protons, right? But there are more protons too that are just grabbed and pumped across.
And then where do the electrons go? For the electrons are just moved.
In terms of the complexes, yeah, the electrons are being transferred from one cofactor to another or one functional group to another inside those complexes. And they're dropping in energy as they do so. And like I said, some of that energy is captured to pump protons. So, but we're not going to, in this course, go over the mechanisms of moving those protons. And I would have to look it up because I haven't gone over it before. Other questions?
Yeah, just to clarify, if it's spontaneous, all of them just have to be in order from the bottom to the top of this?
Yes.
Okay.
So that's a cheat for if you were to write out this half reaction, if you were to flip this one and then add it to this one, right? It's going to be spontaneous. Same sort of thing down here. So when we did pyridine, if I was to flip this one, right, because it starts with lactate, the first reaction is, let's just look at it. The first one is lactate, right? Lactate goes to pyruvate plus fumarate goes to succinate. We can add those together and we're going to get, I'm just going to abbreviate them, lactate plus fumarate goes to pyruvate plus succinate, right? And when I flip lactate to pyruvate, right here, that's right here, right? That's going to be plus 0.185, right, volts? Okay. And then when I don't flip, because I don't flip, I'm having fumarate go to succinate. That's right up here. That's plus 0. That's a 3? Yeah, 0.031 volts. And so I add those two together and I'm going to get 0.216 volts, which is spontaneous because it's positive. So, but if I do that for anything, if I go from here to there, that will always work because of the way the chart's lined up. And I won't mess with your chart. We'll always give you a chart, negative to top, positive on it. That's how they're written. Any other questions before I move on? Okay. So that's electrochemistry there. That's going to come up next chapter or next section when we talk about photosynthesis. But before we do that, remember that what's going on is we pump those protons across so we can actually make ATP with an ATP synthase. So this would be ATP synthase here. Whoops. So ATP synthase is going to let those protons back into the mitochondrial matrix and use the energy of them coming back in to synthesize ATP. All right, so in doing so, it will capture some of that energy and use it to synthesize ATP. Okay. So for this diagram, this is actually the opposite way than we're used to seeing it. The matrix is going to be up here, the opposite way we've been drawing. The intermembrane space is down here. So imagine this being your membrane. And so what we haven't emphasized here, but there's going to be a big difference in protons. There's going to be a bunch of protons out here and not many in here. All right, so we're going to have a difference in the number of protons here. Actually, the way the mitochondrial matrix works in terms of the energy here, do you remember we had our equation for that the free energy for potential across the membrane, right? We have our membrane, its potential is going to be equal to our chemical potential, right? So that's going to be so it's going to be out, let's say out over in. Actually, whether that's a plus or a minus in front of the RT depends on where out over in is. And off the top of my head, I'm not sure well, how we define it. Oops, I'm sorry. I just messed up the notes. Maybe if I don't write anything over there, it'll still be okay. I'm trying not to drag, make it so things split. Plus, here's this thing we just saw, right? The number of electrons, right, or the charge in this case, okay, times Faraday's constant times the potential difference across the membrane. Okay, so there's a potential across this membrane caused by a difference in charge, and then there's also 4 protons. That difference in charge could be protons, it could be sodiums, it could be potassiums, it could be lots of stuff, right? There are lots of things that we could have differences across the membrane that could contribute to this charge bit, and then there's a chemical bit as well. Oops, not the whole thing there. So we could think about this as chemical, so chemical gradient, and we could think about this as electrical. And so you don't need to know this, but I'm just telling you because I think it's fun. In terms of making ATP in mitochondria, that actually ends up being the dominating force. Okay, so you've got other things that got moved across the membrane as well, and you end up with a potential difference that's bigger than the pH difference. If we think about pH, that's not so big a deal. That's not so much of a big a deal. I think the pH difference it's not huge. It's not even 1 unit. Okay. When you go to chloroplasts, you have the same sort of thing going on for photosynthesis. But in chloroplasts, actually this is almost 0, and this part's the big deal. You actually in chloroplasts end up with a pH difference of 3 across the membrane. So you end up with 1,000-fold more protons on one side of the membrane than the other. Right? So nature uses the chemical gradient, it uses the potential. Why it uses one in one case and one in the other, I don't know. Questions on that? That's mostly just interesting. I don't care that you know that. Okay. All right, so we got these protons here. They're outside. They are going to come through this little thing right here. This is like a water wheel. They're actually going to come through this. This is going to spin. It's going to cause this to spin, okay? And it's going to make ATP. All right. Actually, no, I'm sorry. It's not going to cause this to spin. This is going to be held in place by something I'm going to show you in a minute, okay? So, but let's talk about it more. So it's going to cause this to spin, which is going to cause this to make ATP. Oops, ADP. Okay? All right. I misspoke that. I really care that you know this doesn't spin. So let's take a look. We've got a better diagram down here, or a more, I don't know if I say accurate, but because the other thing's actually a crystal structure. So we've got this thing right here on the right here, and this shows it a little better. So what we've got to make ATP, I'm going to write this out. We've got different subunits for this, the way it's labeled. We have these C's, right? You see that? Those are C's. I'm going to go ahead and let's pull this out of the way. Let's make this other thing bigger. And so I'm giving you a kind of rough explanation here. We're going to have a video we're going to watch after that. Okay, so if I look at this, to make ATP, protons are going to go through the C subunits that turn. So protons are going to cross the membrane, moving through the C subunits that turn as a group. So you see how they make a big circle there? The big circle turns. Okay, this turns, do you see this gamma? This turns the GABA subunit. Which pushes, or let's say that pushes against the alpha and beta subunits. Causing them to change shape. The shape changes result in the production of ATP. All right, so in 2 or 3 sentences, however many that was, that's how this works. All right, you see this little B over here? This B subunit is going to hold the alpha and beta so they don't turn. All right, so it's held in place. It doesn't turn. This thing right here is a little offset, and as it turns, it pushes around in here, and it causes these to change shape. And that's how we make ATP. So you've got an enzyme, it changes shape, it binds ADP and phosphate, it smashes them together, it releases them. 3 different affinity states. We'll go over those in detail. Questions? The C subunits are like a water wheel. So protons will come in here and will turn around and will spit out. What was that? It's not momentum, it's concentration gradient. So the wheel is going to not spin we'll see some more pictures. But there's things that kind of keep the wheel from spinning the other way. And then it's spontaneous for them to spin, and it's spontaneous for protons to move from here to here. So that helps it spin the one direction. Let's watch a movie. Now that I've introduced that. We can come back to this in a minute.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. flow through an entry open to one side of the membrane that binds to other subunits.
What was that?
Only protonated subunits
I
can then
guess.
rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
So here's that B thing holding these so you can't turn, right? You see how they're changing shape here?
The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
Which is pretty cool for an experiment to demonstrate that. Okay, so that's a nice animation of how it works.
How does the how does it not get too
Thank you.
wound up if the one part spins and the other doesn't?
How does, what was that?
Like if one part spins and the other doesn't, how does it not just get too tight? I'm not.
So think about it like, I mean, so you have, so this piece right here, right, it's not bound here at all, right? So there's no covalent interactions here. And there's no covalent interactions in here. So this isn't spinning. This can spin inside. Yep. So I'm trying to think of well, I mean, just, you know, like, it's like that.
Yeah.
Right? This isn't gonna, it's loose enough, except that I want something to push and cause my hand to change shape. But, so that's essentially what our mechanism is. Yes? Is that a question or are you stretching?
Yeah, but okay. So like in the so you're saying these subunits hold down the beta subunits and don't transfer them. Is there covalent interactions with that in the membrane at all? Like how
So, covalent interactions where?
the B subunits,
Yeah.
the two big bars.
They're probably, these subunits probably don't have any covalent interactions between the beta and the alpha, right?
That's not the B subunit though.
Oh, the B subunit right here.
So does
This looks like this is probably
so that's
either really strong, this is fixed.
that's like solid in the membrane.
This is solid in the membrane and whatever this A subunit is. So, and whether this mechanically, how it's in there, right? These things assemble, right? They're separate subunits. The reason, if they're labeled as separate subunits, what it probably means is they are synthesized separately. So it's possible that as separate genes, separate proteins, and then this assembles. Okay, and I can't comment on what the connections are here.
Yeah.
If you made me guess, I would guess that they're non-covalent. Although the way this is drawn, it doesn't look like I have very much, right?
Mhm.
But I would guess that these are all non-covalent, but they're pretty strong for whatever reason. Here you do not have strong interactions, or else this wouldn't be able to turn.
Also, what is that? What is
What-
epsilon or
What's the epsilon? I don't know. It's something that helps hold things together. It's something needed to help hold things together.
same with the delta? Isn't that delta?
Yeah, delta looks like it's attaching this to here.
Okay. Do we need to know, like, because in the video it looked like it was only adding into the beta subunit?
Yeah. So what you need to know, the alpha beta subunits work together. These make a domain that makes ATP. So there's an alpha beta, alpha beta, alpha beta.
Yeah.
And each of them is going to be working on its own ATP.
So the video showed that the ADP was only attaching at like 1 subunit. The other one never had attached.
I have to look at that, but when you think about it, you should think about the alpha beta subunits as catalytic units. Okay. So I don't know which is important, but they're both clearly important for it. In terms of, we've had a lot of questions about what's going on here. You just need to know, you don't even need to be able to name this stuff, right? These, I don't care about so much, but remember that it's there and it fixes this in place, so it can't turn, okay? You need to talk about C subunits, alpha beta subunits, and this gamma subunit that pushes against them. That's what's important for you to know in terms of what I'll test on. Okay, let's go down here and look at this. So what we've got, I just want to look at the water wheel and ask you a question. So we've got this little water wheel bit. This is our C subunits, right? This is that thing on the side. What's it, A? And so we have a place for protons to come in, and those protons are going to get here. What's out here right outside the wheel?
Membrane space.
Membrane space, not intermembrane space, membrane, right? It's membrane itself. So we see H plus here, but what do you think those H pluses are sticking to?
Negatively charged amino acids.
Yeah, negatively charged amino acids. Okay, so you can see that this thing is going to wait. This thing is probably going to wait until a proton comes in before shifting over, because if this is a negatively charged amino acid, it isn't going to want to go in the membrane, right?
Mm-hmm.
So proton comes in, it neutralizes it. Now this can move into the membrane, and then it comes back out over here, and the proton can pop out. Yes.
So it can move in both directions, though.
It can move in both directions. Yeah. When I started earlier, and I started to say it couldn't. But yeah, I guess it can, because you could protonate here and then go through, right? And it just depends on where you have higher concentrations. And so what that means is probably, in reality, both things are happening. It's like any double arrow situation there. You're just making a lot more ATP. Otherwise, you're making a lot more ATP under normal conditions. Okay, so let's talk about this from a top view, and then we'll be done for the day. We'll have to talk about some proton counting a little bit later. So what I've got here is I have three different positions here for that. So this is our gamma subunit in the middle. I'm going to make this bigger in a second, but we've got gamma subunit is going to turn, and then get back to where it was. All right, it's got a 120-degree turn of the gamma subunit. As it moves, it pushes against these other subunits and causes their conformations to change. Okay. Overall, that's what's happening. Let's make those subunits bigger so we can see more of what's going on. All right. Let's focus on the, actually the purple. Purple, I'm going to call it a domain. I was just calling it subunits. I'm going to change domain. And that's made up of alpha and beta subunits. Right, a subunit is a individual protein strand or peptide, we could say. Are you guys with me? Yeah, okay. I was getting some faces. So, all right, so we're going to focus on the O subunit, or the purple subunit. The purple subunit. Domain, thank you. Domain has a conformation. We're going to start right here by this top one. That has a high affinity for ADP and phosphate. Okay, so that's this one right here. All right, two, next one down, right here. Guess I'm going to carry this other thing down with me. All right, so two, the gamma subunit turns, let's say, and it's actually 3 protons. Yes, it is 3 protons. 3 protons pass through the C subunits. The gamma subunit turns. 120 degrees. And the purple subunit, purple domain, changes shape such that it has a high affinity for, actually, let's make this one. Such that it has a high affinity for ATP, it thus combines ADP and phosphate to make ATP. Okay? I don't want to hold you guys over anymore, so I'm going to describe the last bit here, and then I'll finish writing it up for the post-it notes. For the last bit here, this would be 3. Okay, what's happening here is that it turns another 120 degrees, and this, what I originally called 1, has a low affinity for ATP, and so it releases the ATP it just made. Okay, so it's 3 protons in, this thing turns 120 degrees, it changes shape, the domain changes shape to something that goes through a process of grabbing ADP and phosphate, making ATP, and letting go of ATP. All right, thanks guys. Have a good one.